Okay. Okay, I would like to thank for the possibility to talk to you this afternoon or this morning or wherever you are in the world. I'm Piero Carninci from the Riken Center for Life Science Technologies, and as the name of the new institute says, we are developing technologies for life science. So, in particular, we have been focusing on transcriptome analysis technologies, and even more in particular to analyze the 5' end of RNAs. The previous technology were the cap trap and full-length cDNA technologies, and then we have more recently developed and redeveloped various versions of the CAGE technology. With this, we have been exploring RNAs, transcriptome, and particularly have insight on the work of novel non-coding RNAs. And I'm going to touch the various aspects here in this introductory slide in the next several minutes. So just to mention the key technology that we are using now, CAGE. So this slide is very crowded, but the key point here is that we are focusing on capturing the 5' end of RNA by making cDNAs. Only the cDNAs that reach the cap site are selected, and then there are various options. We can sequence the product as it is with the Illumina or Helicos sequencing instrument. And in the past, we have been also using a protocol based on PCR and Illumina. We have also been working a lot to automate and to have a kit in order to everybody in the world can reproduce our data. So CAGE and cDNA technology have been contributed to various projects in the past, and we've been annotating the mouse and human genome in previous projects about a decade ago. We have been contributing, particularly with CAGE, to the ENCODE project, mod-ENCODE project, and also other projects I'm going to touch later in this presentation. So, to mention a little bit more in details, our key project, this is the Phantom project, is one of the oldest consortium to annotate the mammalian genomes, particularly we start with the mouse. We start with a large collection of cDNAs that helped also to understand where the genes are also in the human genome. And we produce more cDNAs and start to standardize rules for the notation. And in 2005, using cDNAs technologies and CAGE, we figure out that the most of the genome is transcribed, and there are many non-coding RNAs. We continue using CAGE to prove that just using sequencing of RNA, we can infer regulatory networks. This was published in 2009, Nature Genetics. And now to conclude this overview of the Phantom is the paper that came out, is the series of papers that came out in March, including 2 Nature papers showing, providing a very extensive map of transcription in human and mouse primary cells. I'm going to expand the various aspects of this. Just to summarize quickly the finding that the landscape, what we understand about the landscape of the transcriptome has been changing, because a decade ago we expected something like many protein coding RNAs. However, we found out that there is the large majority is represented by non-coding RNAs. They are not translated. They constitute more than half of the transcriptome. And also, more importantly, there are many groups recently that are doing quite a lot of experiments to prove that they have a function, and they very often regulate the transcriptome or the genome transcriptome RNA stability. Interactive protein have quite a lot of multitude of fractions that unfortunately we don't have the time to explore in details here today, except for some of the examples. Now, we have this technology to profile with very high throughput the 5' end. So what we can ask immediately is what is the complexity of initiation in the mammalian genome? And in a preliminary study, we have been exploring the difference in brain regions of mouse. And in the first set of libraries we explored, we checked the initiation of transcription in various tissues like hippocampus, somatosensory cortex, visual cortex, and cerebellum. And for this gene here, we see that there are preferentially expressed promoters that are very specific for each of the tissues. So this is very important, because if you ask what is the promoter of the hippocampus, or what is promoter of the somatosensory cortex or the visual cortex, they are probably different. And if you look at the regulation of transcription, it's very likely that the transcription factor that bind in one region or the other region are probably different and tissue specific. So this is very important to profile expression that is specific for each tissue. Now, what we can say about how many transcriptions start inside, how many promoters are in the mouse or in the human genome. And we have been addressing this in the Phantom 5. In the Phantom 5, we have been analyzing something like 3,000 samples. Importantly, the collection of samples is very important. So most of them are from primary cells in biological replicas. About half of the sample here on the left are snapshots, so many different cells in triplicate. And this is published. And the unpublished part is here on the time course, where we have been taking 32 of those cells, have been differentiating under various stimuli. And we are analyzing now the dynamics of this stimulation, and we don't have a paper yet on this one. So by analyzing this very large resource, we can now say something about the extent of the initiation site. And also we can have a very broad catalog of expression in 947 samples. And this is just the top of the list of 947 samples in human. And this first gene is the glial fibrillar acidic protein, so very specific for the brain, so it's expressed only in few of those many libraries. This other protein is CD14, again is expressed in some of the immunocells, and is not expressed in most of the other samples. But if you look at the actin beta, it is expressed essentially everywhere. Now we can show those graphs in horizontal orientation. So we show GFAP, and we can see it is restricted to few samples, but the actin beta is expressed essentially everywhere. The median expression is 0 for the GFAP, but is more than 0. In this case, is around 5,000 transcripts per million in the case of actin beta, because this is expressed a little bit everywhere. So we can see, we can distinguish between tissue-specific and broadly expressed genes in this tissue. And then we can also start to ask if the promoter of those genes are the same or are different. So we can plot them, and we can have those genes that have a median of expression that is larger than 1, more than 0. So they are housekeeping genes. And they are usually driven by CPG promoters. And then we have the other groups that is represented here, those that have the median of expression around 0, and which means tissue specific. And those are usually driven by Tata box promoter. Very often they don't have CPG. And if they don't have Tata box, there is some other specific motif. So we can broadly classify the type of transcription by using CAGE. Most importantly, CAGE can provide the transcription at a single nucleotide resolution. So we can tell exactly which base or all the bases that are used in each promoter. And for this, we have analyzed this promoter, the B4GALT1 promoter. And in a set of sample like astrocyte, 3 human donors, 3 biological donors, we can see that the initiation start is very sharp around a single position. Here, there is a Tata box controlling this. But if you look at a CD14 or CD4, again, this is all in 3 biological replicas, human biological replica. Or if you see this cell line that was used in the ENCODE, we can see that the initiation start is much more changeable. It just goes a little bit everywhere, but with some reproducible pattern that are reproducible for each set of biological sample. We can also group them, and we can group the various type of promoters, and we can automatically call the subcomponent of promoter, and we can also measure expression based on each subcomponent of the promoter, bringing the analysis of expression at a different level, at a higher level that was never tested before. And this could not be done possibly both by RNA sequencing or by microarrays, because there is no such resolution to distinguish at a single base with so high reproducibility. So if you really want to see where are the initiation sites on the promoter that are using different tissue, you want to use this analysis for transcription control, CAGE is the perfect tool for this type of analysis. So to summarize this concept, we can now distinguish each subcomponent of promoter and their tissue specificity. Some are CPG driven, some will be data box driven, some will have other motifs, and each one will have some tissue specificity as represented here. And this is not this is just a concept slide. Is not a real promoter. But if you look at expression in this way and ask how many initiation sites there are in the genome, we can tell for sure there are 180,000 initiation sites for which we are very sure. But if you are a little bit more aggressive, you look at lowly expressed, we think that there are could be even 500,000 of such groups that are kind of treatable computationally. So there is a lot of work to do to really understand what they are doing. Some of them are driven by retrotransposon element or other types of elements. This was published, and there is also a website here, and there's a little bit more on the website later. So we can use this now to infer regulatory network in each cell type we analyze, because we have many primary cells here, and that are really very different in quite beautiful collection of cells. We have the CAGE promoter map and also RNA sequence, small RNA sequencing for each one of them. By analyzing promoter, we can see transcription factor binding site, and then see if around those transcription factor around the transcription station, the transcription factor binding site of transcription factors, and infer which set of transcription factor is responsible for the expression in each of the various cell type. We can also infer various type of motifs that are novel, and infer novel regulatory motif for which we still need to find some transcription factor binding site. Because we have RNA-seq as well, we can now also go for transcript discovery and understand gene models a little bit better, and have long non-coding RNAs that are being analyzed in this project. Not everything is published. And this is an overview of all the primary cells and the tissue that we have been analyzing in human. And their relationship, similarity, and differences. It is published in Nature, but if you want to go into the details, please refer to the recent publication. But it's very important to, for instance, as a single example I can bring is the cancer cell line that are represented here in the middle. And it's quite interesting that the cancer cell line don't remember almost anything about their original tissue, but they cluster together as a cancer cell line, which is quite interesting. So, and many of them called cell lines are there. So, please be aware of this when you analyze the public data. And this is the portal of the website of the Phantom. We have quite a lot of visits in the first half and half. We have about 1.4 million of visitors, and I hope that people can still using even more the database to understand gene transcription. Now this is a little bit of diverging from the Phantom, but this is another paper showing the importance of the high resolution identification of initiation site. And this is in zebrafish. This is the ZPROM consortium, which means zebrafish promoterome. We can and we have profiled fertilized from the eggs to the developing embryo, and there is the mid-blastral transition around here, where the maternal transcript are degraded and the zygotic transcript, the embryonic transcript, are being transcribed. This is a dramatic shift, and we start to. First of all, we understood is that there are essentially sharp promoter here, Tata box driven, but there are essentially CPG driven promoters in the embryonic stage. And also we see that all of this is accompanied by dramatic rearrangement of nucleosomes that are essentially random or not well organized in the zygotic transcript, but they become very well organized and rearranged around the zygotic transcription. Having high resolution helps to infer mechanism and the spacing and the complexity of the transcriptional machinery, the RNA polymerase too. Now, I would like to go back to the phantom, and I would like to go back to another finding which relates to identification of enhancer using CAGE data. Now, this was the second article that appeared in Nature, but the key finding here was not our finding, but was a finding from various groups identifying that there is a bidirectional transcription in case of enhancers. So enhancers are bidirectionally transcribed as represented here, but the key point here is that we identify the tissue specificity, the cell specificity, and provide a very large cell specificity for this type of resource. Also, we can correlate very well CAGE with other specific marks, and we can provide very specific function. For instance, we find an answer for the B cells that is not used in CD4 and natural killer monocyte, and then we can extend this analysis to all the phantom sample, finding that there are enhancers that are specific for the various tissue, and they have all the various markers, the hypersensitive site, H3K4, K37 acetylated, H3K4 methyl 1, and so on. And all of this makes a lot of sense in light of what we know about the everything that binds the enhancer. And the key point for those 44,000, we know, for about 60% of them, we know that are quite strongly cell-specific, and we can really use this for a lot of analysis and additional studies, which is not provided anywhere else, tissue specificity. Of course, we validate several of them and have quite a lot of additional data. Of course, we tried the first in cells, human cells, and those have a very high validation rate in various cells. But then we also went in zebrafish, where we have a reasonable success for some of the neuron or neuron stem cell enhancer. The first example is for an enhancer predicted in neurons, neural stem cell, and it makes sense because it is expressed in these areas in the zebrafish embryo. The second example is an answer that is specific for human neuron tissue, and we can see here on the head and on this area here. So this makes a lot of sense that the expression is recapitulated also in zebrafish, which is 3 out of 5 is the frequency that is expected to be validated if you use a human enhancer in zebrafish. We can also infer which enhancer is correlated with which promoter, and this is done by analyzing co-expression. So we analyze expression of CAGE tags at the enhancer and expression of CAGE tags at the nearby promoter, and very often we infer, as in the numbers here, we infer that the correlation, probably what regulates what, and some of those lines with the red line were also confirmed by Chiapet by independent experiment, demonstrating that our analysis can indeed reveal correlation and interaction between enhancer and regulated promoters. Most importantly, I would say that many of the GWAS, many of the SNPs that are associated to disease but not associated to genes, are associated to enhancers. And this is very important because we can put many of the orphan GWAS back to some potential function in enhancer region. So this was quite surprising and quite important aspect. Now I mentioned about non-coding RNA in the early part of the discussion, but we didn't expand so much what they could be possibly doing. But in a recent project, we start to analyze them in IPS, and yes, we prepare a very large collection of CAGE on RNA, particularly nuclear RNA. It's been very important in many replica mouse and human, and tribological replica mouse and human, and using nuclear RNA, not only cytosolic, and the CAGE was being the key technology, plenty of data. The key point is is our analysis, and we found a class of transcripts called NAST, which means non-annotated stem transcripts. Usually they are nuclear, embryonic stem cell, or iPS specific. And those are the NAST, so essentially, particularly in the nuclear, are essentially many intergenic or antisense orientronic, so that they are more than 70% of the transcripts are like this. And we can see that they are below this orange bar, which means they are essentially in the nucleus as represented here. They are also quite specific, so they are, if you compare with the whole phantom, they are only in stem cell, they are nowhere else. And they also carry not only promoter marks, but also enhancer marks. So importantly, we analyze their genomic context, and surprisingly, we found that many of them are not conserved. And also here in blue, they are on retrotransposon element. 40% are on retrotransposon element, and in particular, we found that many of them are on LTR element. This is mouse and this is human, and so which was kind of surprising. Most importantly, then we started to analyze their function. We started to knock down those retrotransposon element derived RNA, and see if our cells shift in the pluripotency by measuring the activity of non-annotated after knocking down those non-coding RNA. And we can see that for a reasonable number of them, we can see there is a shift, and that the cell tend to differentiate, and the marker for pluripotency, non-OG, and then others we validated extensively for 4 of those NAST RNAs. Those are clearly differentiated going down. So pluripotency marker go down, and the cell tend to differentiate in vivo. So many of them are bidirectional transcript also here, like in the previous example for the phantom. So and they are essentially bidirectional transcribed, suggesting that they could be an answer. We've been checking a little bit more in details, and we found both DNase hypersensitive site and P300 suggesting that they are potential enhancer. And because of that, we have been also checking with CHIA-PET. The transcribed LTR are connecting with active promoters, while the non-transcribed are not. And essentially, if you look at which promoter they connected by Go analysis, we find that they are DNA packaging, chromatin organization, or cell cycle. In particular, cell cycle is very important for the stem cell. So we identify retrotransposon element that are an enhancer, and they are connecting with the active genes in stem cell. If you knock down the RNA, the cell differentiates, suggesting that there are some potential very interesting functionality for this. So we published in Nature Genetics quite recently, and so the basic idea is that once upon a time there were the ancient retroviruses that were parasiting the cell, causing damages, but the cell has learned how to adapt them and to adopt them, and take advantage of them, and particularly probably because they can provide a regulatory element, spread the regulatory element in the genome very efficiently. And now those old retrotransposon elements are producing, are essentially regulating cells, acting as promoter, acting as enhancer, and those are particularly important for the existence of and maintenance of stem cell, and they are downregulated during the differentiation of stem cell. And it's not only us, also Professor Yamanaka at Kyoto University has recently found some of those LTR elements that are also regulated and downregulated during the differentiation of cells. We have a little bit more stories about what was used to be called junk, and still many people consider junk, but we are looking at this quite carefully. One is that we found many of those LTR elements, slightly different classes, but again those LTR elements that are very highly expressed in cancers. And particularly they are particularly in liver cancer and in tumors, and they are not expressed in normal tumors or in other types of cell as represented here. So the final story I would like to tell about this is that the final class of non-coding RNA that we've been analyzing. And here we were interested in sense-antisense, so particularly the sense is represented here by this green RNA, and the antisense is this red RNA in the opposite orientation. And this is a gene, UCHL1, that is important for the familiar Parkinson's disease. We overexpress the RNA in cells, and we saw that if you overexpress the RNA, the sense RNA doesn't change at all in expression level. However, if you look at the protein level, we see that the protein level is dramatically enhanced. It's not inhibited, it's enhanced, in the contrary what everybody expects. So those non-coding RNA help the cell to make specifically much more protein. We've been looking at what are the responsible sequence, and we found that by computer analysis, we found that there is some SINB2 element here represented in this green part. If we delete this sequence here, we see that we cannot have any more the induction of protein translation in our cells. So we try to see if you can transfer this property to additional RNAs by changing the antisense region. So we design those RNAs that we call now SINE-up, because the SINE element up-regulating translation. So if you change the antisense region, we change the specificity, and we see that we can also bring this to the GFP, so we can produce more GFP if we change the antisense region and use the remaining part of the SINB2 element. This was again published some time ago, but the basic idea is that we can have a tool that is just the opposite of siRNA or microRNA, and by changing the overlapping region with the various messenger RNA, we can now change the specificity and do various applications. Now in the lab we are very intensively working to explore all of these applications, optimize sense-antisense region, and we can use this in vitro, in vivo, in cells, and there is quite a big things ahead. So I'd like to conclude saying, of course, there's plenty of work ahead. There is plenty of unknown stuff here in this unexplored part of the transcriptome. It's very exciting, and what we better need functional assays, and we need also young scientists also to come and work with us or work independently. But anyway, we need those tools. And in general, because all of this was about finding a function of out of the junk, out of the non-coding RNA, I think it's better to be very open. We don't claim that they have a function, but very often there may be function, and as very important people say that we should be very careful and try to see what the facts are telling us, rather than sticking on the basic idea. I'd like to show a little bit of the Phantom Consortium. I'd like to thank many people for this activity, and clearly I cannot read all of them, but they will stay online, and if you scroll, you can read all the name or look at them in the acknowledgement of the Phantom paper. Is also the ENCODE consortium and additional grants. And we are in Yokohama. If you have some interest, please don't hesitate to contact me. And yeah, this is a list of grant. And I'm open for question. And I think that there is a question and some question online. I guess I must read the question. And dear Piero, is Asutoshi Dingra. I apologize if I don't, Asutosh Dingra. And the question is, dear Piero, greeting from Tübingen, Germany. My question is related to gene targeting. If we focus on making knockouts in iPS, especially using TALEN or CRISPR-Cas9. Can targeting TSS site instead of random sites in the exon for knockout strategy would work better, and also possible to reduce off-target effect? This is clearly a very good point. We didn't do it yet, but I believe that targeting in initiation site is probably going to be very effective. I think that the key point is to identify which part of the promoter is important. If you have a very accurate initiation site driven by data box, clearly you can target that very, very well, and you can eliminate that part very carefully. However, if the sequence is very long, like if you have a long CPG island and you have an initiation site over 50 or 100 or 200 base pairs, you may have to delete the whole regions, I think. So I think that you need to really be quite careful on which part is eliminated. We often see that if you eliminate some component of promoter, some other part of the genome may take over and create a new initiation site. So I think it is a little bit complex. And at the moment, I don't see, I don't have other questions. So I'm still be available here for a minute or two if anyone would like to. Okay, there is another question is from Marion, who is actually he's Marian Balkanin is actually our student here. I see you have a long history of I see you have a long history of development of new tag sequencing protocols. Any plans for new ones? CAGE successor maybe for upcoming Phantom 6 or Phantom projects. Okay, this is clearly we are developing new tools all the time, and in the current development of technology, we have been working intensively to optimize, to standardize, in order to have a very reproducible result over time. This has been one of the very, it's not so exciting, but it's very important. Standardization is very important. The other one is to go on the single cell, and on a single cell capturing as many RNA as possible, or ideally most of the RNAs, going on the single cell. And this is what we are working on with several colleagues, including Sharplessy and his group here at RIKEN. Right. All right. So let me see. Okay, there is no questions available anymore. So I would like to thank everybody for the attention, for the possibility to be here. I hope the video will be available, will be online, and please don't hesitate to contact me, whatever other question you have. Visit us, visit the website, write to me, and thank you again, very much again to everybody. Bye-bye.